Welcome to our new scientific video by BestDX Academy. Today we will be exploring the digital Western blot, a powerful capillary-based immunoassay that has revolutionized protein analysis. Let's delve in and see how this technique works, compare it to traditional Western blots, and discuss its advantages and applications. Now let's begin. What is digital Western blot? The digital Western blot, also known as the capillary-based immunoassay, is an advanced method used for detecting and quantifying proteins in a sample. It is a modern variation of the traditional Western blot technique, which has been widely used for protein analysis in various fields of research, clinical diagnostics, and drug discovery. Like traditional Western blots, it relies on the principles of gel electrophoresis and immunoassay techniques. However, the digital Western blot uses capillaries and advanced imaging technologies to offer higher sensitivity, accuracy, and reproducibility. So how does digital Western blot work? Let's break down the process of a digital Western blot into 5 main steps. 1 is sample preparation. As with any protein analysis technique, sample preparation is critical. The protein sample is mixed with a loading buffer and denatured, which allows the proteins to unfold and become linear. Capillary electrophoresis, shortened to CE, is a critical step in the digital Western blot process, as it allows for the separation of proteins based on their size. This separation is essential for identifying and quantifying specific proteins in a complex mixture such as a cell lysate. In the first step of the capillary electrophoresis process, matrix loading, capillaries made of glass or fused silica are filled with a gel-like matrix. This matrix, comprising a stacking and separating matrix, is ideal for size-based protein separation. The stacking matrix, with lower polyacrylamide concentration and larger pore size, concentrates and aligns proteins. When an electric field is applied, proteins move through the stacking matrix, forming a narrow band. This ensures better separation in the subsequent separating matrix. The separating matrix, with higher polyacrylamide concentration and smaller pore size, performs the actual size-based separation. As proteins migrate from the stacking to the separating matrix, they encounter smaller pores, separating based on size. Smaller proteins travel faster, while larger ones slow down, resulting in efficient, size-based separation. After the matrix is loaded into the capillaries, the protein sample is prepared, and this step is called the sample loading. The sample is mixed with a loading buffer containing a denaturing agent such as SDS or urea, and a reducing agent such as beta-mercaptoethanol or dithiothredyl. These agents help break down the protein's tertiary and quaternary structures, allowing them to become linear and uniformly negatively charged. This uniform charge enables the proteins to migrate through the matrix based on their size and not their charge. The denatured protein sample is then loaded into one end of the capillary. Next, once the sample is loaded, a voltage is applied across the capillary, creating an electric field. This electric field causes the negatively charged proteins to move towards the positive electrode. As the proteins migrate through the gel-like matrix, they experience different levels of resistance based on their size. Smaller proteins can pass through the matrix more easily and therefore travel faster, while larger proteins are slowed down due to their larger sizes. This size-based separation allows for the isolation of individual proteins or protein complexes, which can be further analyzed. In the digital Western blot, immobilization captures separated proteins onto the capillary's inner surface, coated with a chemically reactive substance like hydrogel or polymer. This substance forms covalent bonds with proteins, providing a solid support. Post-capillary electrophoresis, proteins remain in the capillary. Immobilization occurs through covalent bonding between the reactive substance and protein amino acid side chains using cross-linking agents or UV light. Proteins attach to the capillary surface, preserving their separation pattern for subsequent steps like antibody probing and detection. Next is the step called immunoprobing. Immunoprobing identifies and quantifies target proteins within the separated protein mixture by using specific primary antibodies that recognize and bind to their target proteins due to their unique antigen-antibody interactions. These primary antibodies have high specificity, which means they can selectively bind to their corresponding target proteins, even in a complex mixture of proteins. Secondary antibodies are needed to amplify the signal generated from the primary antibody target protein interaction and enable the detection and quantification of target proteins. The secondary antibodies are designed to recognize and bind to the primary antibodies, effectively forming an antibody sandwich complex: protein primary, antibody secondary antibody. The secondary antibodies are typically conjugated to enzymes or fluorescent tags that produce a detectable signal. During the process, unbound primary and secondary antibodies are sequentially removed by washing steps. Detection and quantification. Finally, the labeled secondary antibodies are detected using advanced imaging technologies such as a CCD camera or a laser scanner. The resulting digital image can be analyzed using software to quantify the intensity of the signal, which is directly proportional to the amount of target protein present in the sample. Digital Western blot and traditional Western blot are both powerful techniques for protein detection and quantification, but they have some key differences that can influence the choice of method for a specific application. Here, we compare these two techniques based on various parameters. Separation and transfer. In traditional Western blot, proteins are separated by gel electrophoresis, usually SDS-PAGE, and then transferred to a membrane, typically nitrocellulose or PVDF, through a process called blotting. In digital Western blot, capillary electrophoresis is used for protein separation, and the proteins are immobilized directly on the inner surface of the capillary, eliminating the need for membrane transfer. Sensitivity. Digital Western blot is generally more sensitive than traditional Western blot, as the capillary format allows for better signal-to-noise ratio and detection of low abundance proteins. The enhanced sensitivity of digital Western blot is particularly useful for detecting proteins present in low concentrations or when sample amounts are limited. Quantification. Digital Western blot enables more accurate and precise quantification of protein levels compared to traditional Western blot. Traditional Western blot is often semi-quantitative, relying on band intensities which can be affected by various factors like transfer efficiency and antibody binding saturation. Digital Western blot offers a more linear and reliable quantification, as it directly measures the fluorescence or chemiluminescent signal from the antibody-protein complex. Reproducibility. Digital Western blot offers better reproducibility than traditional Western blot due to its automated nature and minimized manual handling, reducing the potential for human error and variability. Time and labor. Digital Western blot can be faster and less labor-intensive than traditional Western blot. The capillary-based system streamlines the separation, immobilization, and detection steps, reducing the overall time required for the experiment. Additionally, the automation of certain steps, like capillary electrophoresis and immunoprobing, reduces hands-on time for researchers. Sample consumption. Digital Western blot generally requires less sample compared to traditional Western blot, as the capillary format allows for more efficient use of sample material. This can be advantageous when working with limited or precious samples. In a word, digital Western blot offers several advantages over traditional Western blot, including increased sensitivity, better quantification, improved reproducibility, reduced time and labor, and lower sample consumption. However, the choice between these two techniques depends on the specific application, available resources, and the desired level of accuracy and sensitivity for protein detection and quantification. Thank you for watching.